Welcome, everyone. Good to have you here. This is a preprint that was posted by Rick Betzel and his colleagues. Rick was a graduate student at the same time I was at Indiana. He was a graduate student in Olaf Sporn's lab, and he is, not surprisingly, an expert in connectivity and graph theory. He's now working at the University of Minnesota. And I remember I was at OHBM a couple months ago in Australia, and I saw Rick at one of the poster sessions, walked up to him, and he was kind of sighing and shaking his head. And I said, Rick, what's wrong? And he said, well, it just seems to get more and more abstract. He was speaking specifically of a lot of resting state, a lot of functional connectivity, a lot of graph theory posters. And I think that this preprint is his way of trying to get us back into a more concrete space and think about, even though we have all this graph theory, more abstractions about what resting state connectivity and the correlations can do, we need at some point to have a biological interpretation. So that's the focus of this paper. I'm going to begin by giving a brief review of functional connectivity, the so-called mesoscale, which Rick talks about in the preprint, because this underlies a lot of his arguments about why we need a finer scaled interpretation of what these connectivity values and correlations actually mean. If we go back to 1995, which is now 3 decades old, it's quite astonishing to think about, but Brat Biswal was doing some research. I believe this was primarily about trying to observe physiological noise in the data set, something like that. He stumbled upon this. This was one of those serendipitous discoveries that happens to move scientific fields along. Although, fortune tends to favor the prepared mind. He did see something, and then he thought, that's kind of strange, and he delved more into it. And what he saw was this. If you see on the left in panel A, a typical motor task could be pressing left buttons, right buttons, any kind of motor task, something that very robustly activates motor cortex. Not surprisingly, we see that's very, very clear. But he also noticed that if you had somebody really do nothing at all and just lay in the scanner, you could average across a region. Let's talk of a region as simply a collection of contiguous voxels. You could average across the time course in that ROI, region of interest, and then see where that average time series correlates with the time series of other voxels or regions in the brain. So you can see here, if we take B as a seed region and average across all the voxels in this circle here, you get a strong correlation with the opposite motor cortex, which, if we think about the functional organization of the brain more broadly, this could make sense. Maybe the motor cortices are always sharing some kind of overall time series, some kind of overall pattern of activity, because usually motor responses from the left and right parts of the brain, like walking, using your hands, they need to be coordinated. So maybe they're always somehow communicating with each other, whatever that may be, and that this correlation is a reflection of that. So for example, if we take the time course where my crosshairs are right here in a functional brain image, and we look at the overall time series, which is plotted in blue, and then we look at some other distinct region of the brain and plot that in red, you can see that it's not a perfect correlation, but the correlation's pretty high. So it's simply, usually when we talk about functional connectivity, we're just taking the correlation in time series between one region and another and calculating some kind of Pearson's correlation coefficient between time series. And this, not surprisingly, it's a very robust network. This is the so-called default mode network, a pattern that's very robust and you see across many different kinds of resting state studies. It's also worth clarifying some of the language, talking about types of connectivity, because some people do take issue with this mesoscale, this functional connectivity, as not really being connectivity per se. So you have some diehard purists who prefer to call it bivariate correlations. I'm not making fun of them. I do think that they have a good point. For better or for worse, we're saddled with the label functional connectivity. People just know what it means. It's very well established in the literature. And the reason people may take some issue with this is that when we talk about connectivity, think about the wiring in your stereo systems, your computers, your cars. It's very clear one cable connects to another and has some kind of effect on a motor or an engine, something like that. With functional connectivity, you're simply observing correlations between two or more different parts of the brain. And these correlations, which we know from extensive literature critiquing functional connectivity, these can be driven by spurious effects. Could be artifacts like motion, that's a big one, physiological artifacts, respiration, heartbeat, anything that systematically can change the position of the head ever so slightly, but still consistently, or leading to some kind of systematic change in two or more distinct regions. But in any case, this is functional connectivity. Something that seems to make more sense is structural connectivity, at least from a pure connectivity perspective, in which we reconstruct neural pathways, these heavily myelinated white matter pathways that connect distinct regions of the cortex. So usually we use diffusion-weighted imaging to try to reconstruct those. And then we also have effective connectivity. You can think of this as SPM's dynamic causal modeling, in which you try to determine whether the activity in one region of the brain has a causal effect or modulating effect on the activity in another distinct part of the brain. It's difficult to do. I tried this once a long time ago. I didn't get a significant result. Maybe it was due to my technical limitations. Maybe the data just didn't have a dynamic causal effect, unknown. But there is this particular method available, developed by Carl Friston's group in London. So with that out of the way, functional connectivity has been very useful for trying to determine whether there are differences between, say, clinical populations. If we look at this paper by Gracious et al., 2004, you see, if we look at that canonical default mode network, is it systematically different between, normally developed adults and people who might have schizophrenia, bipolar disorder? How are these different? What can that mean about potential therapies such as pharmacological interventions, neuromodulation to try to get those default mode networks? If we try to get them looking more like a control population, could that alleviate symptoms, for example? And another reason for the popularity of functional connectivity is that it's very easy to do. For better, for worse, it is very simple because you don't have to design a task. You might have a fixation cross, the person looks at that, and then you can calculate functional connectivity maps from that. So you don't have to worry about control conditions, really. You do need to worry more, in this case, about possible confounds. Resting state is much more sensitive to those, but it is quite an easy paradigm to include in your experiment. Sometimes, kind of thoughtlessly, Stephanie Noble has written a lot about how we need to think about the overall amount of time we want in order to establish robust connectivity maps. But in any case, they are popular. It's easy to add 5 or 10 minutes at the end of your scan, hopefully more, but really it's very simple to do. And then when we have these, we can parcelate these brains into nodes, regions of interest, nodes. I'm going to use those somewhat interchangeably. But the idea is, let's say we have an atlas. We've normalized all of our data to a standardized template, and we have some kind of motor mask. We have parietal masks, temporal masks, and little subdivisions within those as well. We can create these connectomes, these maps showing the connectivity of every region with every other region of the brain. And it's up to you what kind of scale you want. It could be, in this case, around 80 ROIs by 80 ROIs, could be hundreds, could be a few dozen, could be very fine-grained, could be a little bit coarser-grained. But these connectomes, these profiles, can then be used for graph theory metrics. You can use different kinds of clustering. You can compute different kinds of centrality measurements. You can see what nodes seem to be more connected to a wider range of other nodes, what happens when you remove it, and so on. So everything I've talked about so far is mesoscale. Okay, we're not at the level of neurons. We're not looking at the entire brain, but we're parcellating it into some different regions. But relative to the scale of a neuron, these are like continents. Other issues are we're averaging across time, across individuals, and unfortunately, in a lot of cases, across noise, although we try to control for that as best we can. The nodes themselves may not correspond to functionally coherent units. We talk about individual neurons. It's very easy to understand what those mean. But if we start looking at a gigantic ROI that encompasses billions and billions of neurons and possibly trillions of synaptic connections, it really becomes difficult to say that this is a functionally coherent unit that just does one thing. And lastly, it can be impossible to compare against ground truth. These non-invasive techniques we have with fMRI, EEG, diffusion imaging, they're extremely useful because we now have access to virtually anybody who's willing to go inside a scanner. We don't require open brain surgery. It's not invasive. There's no injections, no radioactive tracers. But when we try to compare it against some kind of ground truth, we run into a wall. Even reconstructing diffusion tracts, these white matter tracts may look very convincing. They may make sense, but they are reconstructions. They're somewhat abstract and a little bit removed from the ground truth. Compounding that, and this is something I show in all of my intro to fMRI courses, when we think about these functional connectivity maps, we do need to take a step back and think about what they're based on. This is a model of the so-called hemodynamic response function, HRF, where if we give somebody an impulse, this is what the overall shape looks like. Now, when there's a task-free environment, these look more convoluted. They're not really responding to any distinct impulses. The mind can wander, for example. There may be random memories and random thoughts entering the brain. But in any case, we are several steps removed from the ground truth of what we want to look at, because we have some kind of stimulus or some kind of mind wandering that's leading to some kind of neural firing. More neural firing needs more oxygen. Oxygen leads to more blood flow through neurovascular coupling. That in turn leads to greater measured signal or fluctuations in signal. And in this case, the observed signal is a few steps removed from actual neural activity. So that's what we're all familiar with. Most of us are familiar with. But let's get back to a finer grained scale. So now we're looking at individual neurons. This is a photo credit. This is Man at Mouse from Canva. Some of these are freely available. I just want to give credit where it's due. I have no idea who that person is, but this is a pretty nice figure of the neuron. So just to refresh your memory, we have presynaptic neurons and postsynaptic neurons, some kind of synaptic cleft through which neurotransmitters are sent from one neuron to the other, have some kind of excitatory or inhibitory function, depending what the neurotransmitter is. So these can be neurotransmitters, neuropeptides, primarily inhibitory ones like GABA, excitatory ones like glutamate. Many things are happening at this particular juncture. So that is really, it has a lot of biological meaning. We can get a really good sense of whether these neurotransmitters at these synapses are leading to a greater activity or depressing activity once they cross that cleft. And if we look at this scale, now we're going into nanoscale, where we're talking about the terms used in this preprint. Each edge, okay, this is a graph theory term. So you think of an edge as usually a connection from one node to another. Okay, think of that as a synapse. This gives you a better idea of discerning directionality. You have a good idea of what effect one neuron is having on another. And once we have that, we're able to study the interaction of connectivity with neurotransmitters. So instead of at this mesoscale thinking about just the correlations between time series, which as we've seen is a few levels removed from actual neural firing, here, if we can have direct measurements of one neuron and the connecting neuron, we have a very good idea of, first of all, which neuron fires first, what's the temporal sequence, and what's the effect on the downstream neuron. What is it doing? This is figure 1 from the preprint paper showing the different scales that we're working with. Apologies for the resolution. It's a little blurry. I had to blow it up a little bit. So up here in the upper left-hand corner, this is a diffusion reconstruction where we have diffusion images. We reconstruct something to get these whole brain chektography maps. And this, again, is done at a relatively coarse scale. Okay, even if our resolution is sub-millimeter, these voxels still contain millions and millions, perhaps billions of neurons. Probably not billions, but a lot of neurons in each voxel. On another level, in B, we have stereotaxic injections in which you have some kind of tracer, but this usually requires a post-mortem dissection to see exactly where this was diffusing throughout the brain. On the lowest scale, or the highest resolution scale, I should say, you have nanoscale imaging, in which you can use something like electron microscopes to actually see individual neurons and individual synapses. So the spatial resolution of nanoscale, it's a few orders of magnitude or more finer resolution than what we're used to with a typical diffusion-weighted or T2-weighted image. And in this case, we have better idea of weighting, about direction, about neurotransmitters or neuropeptides that are being sent across the synaptic cleft. Another issue comparing mesoscale to nanoscale is with mesoscale imaging, like a typical functional connectivity map. Let's say you have two regions, parcel A, parcel B, shaded in red and blue. It could be multiple things that are leading to an observed functional connectivity between them. So for example, if we look at a closer scale within each of these, let's say, just for the sake of argument, there's 4 neurons in each of these ROIs. Obviously not at this scale, but let's say there's 4 of them. It could be one of the reasons we're seeing this connectivity between them. Maybe every neuron in one of the parcels has a direct terminal or terminal neurons, some other neurons connected to in the other region as well. Or it could be that maybe only one of these neurons is connected to another neuron and the other one, but there's a lot of modulating activity among the neurons in one of the parcels and the other parcel as well. And then this leads to some kind of communication, some kind of directionality between them. However, at the mesoscale, it's impossible to resolve this. We have no idea what kind of pattern there is within each of these that's leading to one of these parcellations having some kind of effect on the other parcellation. So over here on the far right, we have both cases appear identical at the mesoscale. And for those who are more familiar with fMRI, think of this as you have a contrast, A minus B, and you just have the result of the contrast. You have no idea whether it's because A is a lot greater than B, or maybe they could be both negative, but A is less negative than B, but they're leading to the identical contrast that you're observing. And it's always a good idea to unpack those. So in this same scenario, we're trying to unpack what this functional connectivity map looks like by resolving it at a finer resolution. It's also worth talking a little bit about the very basics of graph theory, because this also comes up in the preprint about how we can use it to talk about directionality, about paths between different areas. And a path is a sequence of edges that connect nodes. So at the mesoscale, functional connectivity, the nodes are those ROIs I was talking about. The edges is the Pearson's correlation between them. And they could be right next to each other. They could be very functionally far apart from each other, but that's the edge. Now, a path is a sequence of edges. You chain them together. One edge leads to another node, then another node, then another node. That's some kind of path, a unique set of edges that it needs to traverse in order to connect two nodes. And a walk is similar to a path, but it allows repeats. You can go back, then forward again. You can traverse the same edge multiple times. Whereas with paths, there's a unique set of edges that you can traverse. Now, with nanoscale measurements, looking at individual neurons, this allows you to determine the directionality of neural firing, as I talked about before. So, in contrast with functional connectivity, it's impossible in most cases to determine whether one region was firing first before the other. Maybe they're both firing at the same time. Our temporal and spatial resolution is too low to make any kind of claim about directionality. There was a well-known, I don't want to really say debate, but critique of something called Granger causality analysis. It's a way of trying to determine, let's say, directionality. But when applied to fMRI data, the temporal resolution in most cases is so low, usually 2 to 3 seconds historically, that it's really difficult, if not impossible, to say that one preceded the other. We're sampling this very slow, sluggish, slow to unfold hemodynamic response function, and it did seem to give a lot of spurious results. So it has fallen out of favor, Granger causality. And I think that if you try to apply it at the nanoscale with neurons, you have a much higher success rate. Now, just a couple more things about graph theory basics. When we talk about these nodes, edges, think of these again, the nodes as the individual ROIs, the edges as the connectivity between them. And you can recast it into a more abstract space. So node 0 may have a connection to 3 and 2 and 1, but node 2 can only get to node 3 by first going through node 0. And again, you can represent this different ways in this graph over here, or what's called an adjacency matrix with ones and zeros indicating whether there's a direct connection between two of the nodes or not. And any network can be represented as a collection of nodes connected by edges. So we get to, again, a more abstract space. We start talking about networks. We talk about what's the overall connection of these different nodes with each other. And usually with fMRI data, we have some kind of cutoff where we say, above a certain correlation value, we assume that those two things are connected. Below that, they're not connected. Sometimes it's helpful to recast it in something like the Amtrak train system, right? So if, for example, I'm in Cleveland, I'm trying to get to Seattle, right? If it's the easiest, it's the most efficient to go through Chicago, then Milwaukee, traverse the Great Plains, and get to Seattle. If Chicago, however, is not working, there's construction, the train system is not functioning there, then I would have to go through a much more circuitous route, basically go along the eastern seaboard and then somewhere along the south before I could get to Seattle. And that would make it much less efficient. So by recasting it like this, and if we were looking at something like a lesion study, somebody has a lesion in one part of the brain, and we know from control subjects that there appears to be a hub there that's necessary for coordinating motor activity, for example. If we remove that, it could lead to inefficiencies, ataxia, all kinds of different motor problems and disorders. The last thing about graph theory I want to mention is that we conceptualize these different clusters of nodes as modules. The simplest way to think about this is the lobes of the brain. We all know about the frontal, parietal, occipital, temporal lobes. That's psychology and neuroscience 101. You'll learn it at some point. And they do seem to have pretty clear anatomical boundaries and pretty clear functional differences. For example, temporal lobe is very strongly correlated with auditory processing. You lose your temporal lobes, probably you're not going to be hearing so well, in addition to things like memory and so on. In visual cortex, the occipital lobe, mostly responsible for visual processing. You lose your occipital lobe, you're probably not going to be seeing too well. So we think we can take that at a very gross scale and conceive of those as different modules. And that's when we say the density of intramodular connections is greater than intermodular connections. You may have read Jerry Fodor's modularity of mind. This is kind of going along a similar a conceptual route. However, it's very important to note that there are both intramodular and intermodular connections. It's not as though the occipital cortex is completely divorced from the frontal cortex. We know that there are very long range connections and also indirect connections between all the different lobes of the brain. It's a very highly interconnected, densely interconnected organ of the body. But this can make it a little bit easier to grasp the overall functional architecture of the brain by recasting it in modules. And within graph theory, no need to get into the details, there are different ways to determine whether something is a module or not. And part of this is your approach, your thresholds of when intramodular connections are greater than intermodular connections. But usually, backed up by theory, backed up by anatomical measurements of the brain, you can get a certain modularity that makes sense for the scale that you are looking at. And if we go from there to the individual neuronal level, okay, modules are useful. Okay, anything that we can use to determine the functional architecture of the brain is useful. But if we get to a finer scale, we start looking at individual neurons in, say, the Drosophila fruit fly or C. elegans, that ringworm, both highly studied animals because they're extremely small and their nervous systems are not too complex, too big relative to other animals or humans. We can get from kind of a coarser scale, looking from flow to superclasses to smaller classes and ultimately to cell types. And what I'm going to be talking about on the following slide is there kind of a halfway point that we can meet from functional imaging and neuronal imaging? Because the tools we have right now, I don't know that if there's a way to non-invasively image every single neuron of a human brain. It's just, in my view, it's just not possible at this point. But if you look at these smaller animals, the Drosophila fruit fly, for example, you can start to reconstruct an entire neural system and also have a good sense of the different cell types, how the neurons are organized, and you can apply modularity in that context as well, but it would have more biological meaning, more biological relevance. Now, just one slide here on layer fMRI. This is a relatively new advance in functional imaging, in which we acquire functional images at a sub-millimeter resolution scale. Now, this requires typically much higher field strengths, like 7 Tesla, for example. I do know that some people, including Luis Hernandez Garcia here, we're trying to see if this can be plausibly done at lower field strengths, like 3 Tesla, but remains to be seen. In any case, the gold standard seems to be using 7 Tesla, possibly higher, to get the required signal-to-noise ratio because SNR increases as a function of field strength. So you'll need both that and a higher spatial resolution to start to resolve individual layers within the gray matter, right? So we know that in the human neocortex, typically we have 6 distinct layers, right? Gray matter thickness is around 2-3 millimeters, maybe more, maybe a little bit less, depending where you are. But if you have sub-millimeter resolution, you don't have 2x2x2 or 3x3x3, you can start to look at individual layers. So what I'm proposing is that possibly, if to try to get closer to the scale that Rick Betzel is talking about, layer fMRI may start to get in the direction of more biologically meaningful functional connections. I don't want to claim too much. Obviously, this is a very far cry from individual neuronal firing. We're still looking at an epiphenomenon of neural firing. But I do think that layer fMRI has the potential to get to a finer scale that may make more sense. If we're looking at individual layers, we do know that layers happen to be composed of similar cell types, for example. They're oriented and structured in a similar way. And that may in turn give us greater insight into their functional organization as well. So we're not looking at huge clusters that may span functionally heterogeneous areas that may bridge different parts of cortex, maybe leak into white matter and CSF, for example. And I think that this is a promising avenue to look at. In any case, what they talk about in their discussion is that obviously we should use both if possible. For the nanoscale measurements, if we can't use them in humans and we're only looking at them in things like the fruit fly and C. elegans, they can still be used to try to create these graph theory metrics, these modules that are biologically meaningful at these very small scales. And if we can see similar motifs, similar patterns of activation at the mesoscale, maybe using much higher resolution fMRI, it could give us some insight into how the human brain is organized as well. So this is another way of saying that there's a potential to bridge theoretical and experimental neuroscience, especially if we have these measurements from both contexts. It could suggest certain principles of communication. And I'm quoting from the paper here, such as redundancy, routing efficiency, or controllability that may be conserved across evolution and scale. So it remains to be seen. Again, this is really a think piece. They haven't presented any empirical studies and evidence so far to really try to combine the two, but there are, I do believe that there are experiments underway trying to combine both of them. But in any case, I do think that it is an important step back from the more abstract way that we're used to thinking about functional connectivity, and to get us more grounded in the concrete details, because the abstractions, if there's no real connection at any point to recover concretely what these functional connectivity measures mean, they're really not going to be too useful. At some point, we do need to know or have a good idea what these mean exactly, and what are the implications for the functional organization of the human brain. At this point, I want to open it up to a broader discussion. What did you think? It wasn't a very long paper. Again, they covered some of the basics of functional connectivity, about nanoscale imaging, about the bridges between the two. And I'd just like to get any thoughts, possibly if there's anybody here who has actually worked with single cell recording, what do you think about their recommendations? Has anybody here worked with layer fMRI? I've only dabbled a little bit with it. I'm trying to use it more. They do have some open access data sets, some 7 Tesla data, I believe, that you can use for. There's a package called LANI. I hope I'm saying that right. LAYNII. This is a layer specific fMRI analysis package. And if anybody's used that, I'm very curious to hear what your experience is like.
Yeah, Andy, I think Max was going to try out the Laney on Luis's data.
Yeah.
So yeah.
Mm-hmm.
But yeah, no, I haven't touched it myself. I've just seen like Palomini's layer work,
Mm-hmm.
where they do able to show some input output connectivity right to the different layers.
Yeah.
So
Mm-hmm. Yeah, I know, Peter Bannattini obviously is a very big proponent of it. And I was talking to Luis yesterday about how he's approaching it. I mean, it remains to be seen, can 3 Tesla give us something that we could use for this kind of resolution? We don't know yet, but it would be very useful if we could make it much more accessible. Any other comments, questions? Well, we can leave it at that. Get done a little early today. Again, some food for thought. We'll see, when, where this gets published, and what some of the follow-ups will be, because I see where they're going with it. I see what they're saying about trying to bridge the two. Um, I'd like to see what some of this looks like when we actually have a really good sense of how the nanoscale from some of these animal studies translates into comparable, you know, functional connectivity analysis, whether those two things really do seem to be on par with each other, or is functional connectivity just really different somehow? Because like I said, that it is such an abstraction in a lot of ways, and we do tend to add more abstractions on top of abstractions, especially the farther up you get into really complex graph theory. All right, everyone. Well, enjoy your weekend. We'll see you next week, and enjoy the start of your semester. All right. Thanks, everyone.